Good morning, everyone. We're very excited that you've joined us today, either live or after the fact by recording. I'm just going to wait for a moment for a few more people to join, and then we'll get started. Okay, we're at a lot of participants, and it's slowing down a little bit. So that may be our cue to get started with this wonderful presentation. And I'm really excited to be here today. Welcome to TMA's Myositis Research Insights, and with special thanks to our webinar sponsor, Bristol Myers Squibb. I'm with some people from BMS today in another place physically. I'm Jane Myles. I'm a member of the board of directors for TMA and really delighted to serve in that capacity after a career in clinical research and drug development. So this webinar series is designed to connect patients and their families to specific myositis researchers in our community. And before we get started, just so you know, the webinar is being recorded and will be available on TMA's YouTube channel as of the 1st of December. We'd love to find out where you're joining us from today, so put your state or country into the chat. And if you have any questions for our researcher today, put them in the Q&A box at the bottom of the screen. We're pleased today to discuss the research of Dr. Begum Horuluoglu. Dr. Horuluoglu is an assistant professor in the Myositis Research Group at the Division of Rheumatology in the Karolinska Institute in Sweden. She finished her PhD in molecular biology in a partnership program between Bilkent University in Turkey and the National Cancer Institute, NIH, in 2009. Her PhD work focused on investigating novel therapeutic approaches for autoimmune disorders such as lupus, myositis, and vasculitis through regulation of immune cell differentiation. In 2019, she joined Professor Ingrid Lundberg's research group as a postdoctoral fellow, where she started working on understanding the immunological mechanism of myositis through molecular and bioinformatic approaches. Whew, that's a mouthful. Since 2023, she's been an assistant professor, continuing her research on the understanding of myositis. Welcome, Dr. Horuluoglu, and thank you so much for joining us today.
Thank you. I'll just share my screen. Yes. Thank you. Hello, everyone. Thank you, Jane, for the nice introduction, and to the TMA board for inviting me to present my research here, and all of you for linking up to listen to me. So today I will talk about my research on T cells. T cells are a part of our immune system, and they help eliminate the invaders in our body. However, in situations like autoimmunity, they start attacking the proteins and cells in our own body, which makes us wonder if they are the allies or are they the enemies in this disease. So before going deep into the research, I want to introduce to all of you the constellation we have at Karolinska to highlight how many people are involved in our research and how interdisciplinary it is. So it all starts when we have our patients come to our myositis clinic at the Karolinska University Hospital, which is composed of research nurses, physical therapists, rheumatologists, which all work together to be able to diagnose the patients and give them the proper treatment. And once the patients give consent, they try to diagnose by taking blood samples and muscle biopsies from the patients. So once the patients give consent to participate in research, we also receive some samples donated for our research. And these come to our core facilities where we have technicians who take care of our samples and put them in our biobank. And here I really want to highlight the importance of biobank and registries because it helps researchers like me who does molecular research and also researchers who perform register-based studies. Without a biobank, it's really difficult to perform any kind of research. And the biobank we have at the Karolinska Institute was founded by Professor Ingrid Lundberg, who is also the leader of this research group. And we have clinical data and patient samples from over 600 patients. That has been collected in the span of 20 years. And it's extremely useful for us to develop new research projects and plans to try to understand the disease mechanisms. And with that, when we have a research plan that we want to perform to understand the disease mechanism, we use this registry to find out what kind of samples we have and what kind of clinical data we have. And our research group is located at the Center for Molecular Medicine, and it's composed of PhD students, postdocs, research assistants, bioinformaticians, geneticists, team leaders, and professors, and which can be both molecular biologists and also clinicians. So after we perform our research projects, then we come all together, including our patient representatives, to understand the results of the data and try to interpret it to be able to help the patients to provide insights to develop better treatment approaches. And last but not least, we need funding to perform all of these steps. And we are very grateful to all our funding agencies, including the Myositis Association, making it possible for us to do research. So after this introduction of our big network and very interdisciplinary network of our research group, let's get back to the science. So in healthy conditions, the immune system, we all know that the immune system is there to distinguish the self from non-self. However, in the autoimmunity, for some reason, the immune system misidentifies the self as foreign and starts attacking it. And if we think of more on a T-cell context, and I got some help from ChatGPT to try to imagine the T-cells in our body in a more daily examples. So if we imagine our body as a large city and each cell type in the city has a specific role. T cells are like our community's security guards. So they go around, they travel the streets, they check everyone, try to make sure each cell is where they belong and it's not causing any harm. And when a T cell spots another cell that doesn't look right, maybe it's an infected cell or a damaged cell, it will either remove it or will call for backup to make sure the community stays safe. But just like every security system sometimes triggers false alarms, the T cells can also sometimes make mistakes. And in these cases, they might identify some of our healthy cells or healthy proteins as dangerous and start attacking them. And this is when we get problems with autoimmunity. So the project that I will talk about today will focus on identifying these T cells that make mistakes. And then if we think of the T cells in the context of myositis, probably you all know this, that sometimes in the muscle biopsies, we can detect these T cells. So here on the left side, you see a muscle biopsy from a myositis patient. And then the brown dots here are the T cells, whereas on the right side, we see a histopathologically normal muscle where there's no T cell infiltrates. So this brings up some of the questions that we have in our group. What are the types of these T cells that are inside the muscle? Are they there attacking the muscle, or maybe they're protecting the muscle from something else? Can we find an indication in the blood that will make us understand what's going on in the muscle? And can we target these T cells to be able to provide new treatment approaches for patients? So to answer some of these questions, we chose a strategy which allows us to investigate each T cell individually. And the best way to describe is this famous smoothie versus the fruit bowl analogy. So let's think that you're at home and you made yourself a smoothie and you're drinking it, but you feel that like there's something off with the taste of it. But you don't know if it was maybe the banana that went wrong or it was a moldy strawberry. So you can't really say when it's a heterogeneous mixture, you don't know which fruit has gone bad. But if you look at the fruit bowl before it goes to the blender, you can clearly see which fruit is moldy or has gone bad. So we have a similar problem when we are doing research. If we think of each cell in our body as a fruit in a fruit bowl and the tissue as a smoothie, we can't really tell when we look at the tissue which is the cell that has gone wrong there. But if we are able to look at each cell as in the fruit bowl by itself, then we can understand maybe which cell has making a mistake. So what we do is we perform a deep genetic analysis on the fruits in the fruit bowl on the single cell level, and this is called single cell sequencing. So during this project, we worked in sync with our myositis clinic that I introduced before to you. So we had patients that come into our clinic with a probable myositis diagnosis. So once they were taking the muscle biopsies to perform the diagnosis, we also received a small piece to perform our project. So when we take the muscle biopsy and then we try to extract these T cells that I showed you with the brown stainings before, then we take these T cells and put them one by one into these small wells. And then we look at we extracted their genetic materials, and then we did a sequencing on the genetic material to understand the characteristics of these T cells on a gene level. And this was done at our core facilities. So once these genetic material was sequenced, we received the sequences, and then we did a computer-based analysis, which is referred as bioinformatic analysis. And we tried to interpret the data. So in this project, we didn't focus on a single subtype of myositis. Rather, we included all the myositis subtypes that we could extract enough T cells. So in the end, we ended up with a heterogeneous group of patients. So we had 2 patients with immune necrotizing myopathies, 1 patient with anti-Jo1 positive antisynthetase syndrome, 1 dermatomyositis patient that was seronegative, 2 inclusion body myositis patients, and 1 patient that was later diagnosed as undefined myopathy, which we kept to have as a control. And it's important here to point out that from 2 of these patients, 1 with the immune necrotizing myopathy and 1 with the antisynthetase, we received samples 9 months after treatment, and the plan was to look at the T cells and their characteristics. What happens after 9 months of treatment? So next, we did our genetic analysis with the computer-based analysis to group the T cells that have similar characteristics or profiles, we can say, together. So for example, one of the most common T cell profile is this effector T cells, which is also called killer T cells. Their role is to find all the infected cells and kill them. So here on the right side in this colorful dot plot, each dot is a T cell. And if they have the same color and they're close to each other, that means they have similar characteristics on a gene level. So you can appreciate that we have detected multiple different groups of T cells. And I want to focus on this group of T cells that we identified in this light pink and dark pink colors, the green and the purple. These T cells are called tissue-resident T cells, meaning that these are the cells that stay in the muscle. And I found this illustration from the Faber lab that really nicely shows what is a tissue-resident T cell or a TRM. So we know in multiple organs like the spleen, lung, or lymph node, we have these TRMs that are the stay-at-home T cells, whereas the effector T cells that I showed before with this killer T cell phenotype, they are the travelers. They go around the veins and then find the infected T cells. So we know in different organs these TRM cells were identified before, but our study was the first to find them inside the muscle. And if we look at, so we had a heterogeneous group of patients, a heterogeneous group of myositis subtypes. So if we look per subtype, what is the rates of each group of T cells? Do they have differences? So here in the bar plot, each column is a patient, and each color is the groups of T cells that I've shown before. You can see that in each of the bars, we see the same colors, but at different frequencies. So regardless of the diagnosis, we saw that all T cell groups were present in all of the patients. then I previously talked about the T cells recognizing invaders, and they do this through their barcode scanners. So if we think of each of any cell that the T cell interacts with has a barcode, the T cell will have a barcode scanner, and we call them T cell receptors. So in our bodies, we have around 400 billion T cells and 1 million different T cell receptors. So it's very unlikely in randomly two T cells will have the same T cell receptor. But what happens is that when a T cell recognizes a target and the barcode scanner scans the barcode correctly, then it will multiply to be able to sustain this infection completely. So if we have two T cells that have the same T cell receptor, that means that they have multiplied because they recognize a mutual target. So we looked at these T cell receptors in our data set, and we saw that in the muscle infiltrating T cells, there was T cells with the same T cell receptor, meaning that there is a mutual target that they have multiplied against. And very interestingly, when we looked at the patients where we had the follow-up samples the 9 months after treatment, what happens to their T cell? Do they have the same characteristics? Do they have the same T cell receptors? Very interestingly, the T cells in the muscle of these patients remained with the exact same characteristics and the same T cell receptors. So here we have two pie charts, which the left one shows the early diagnosis, the first time point, and the second one shows after treatment. And each slice is a T cell receptor. For example, there we found 9 T cells that had the same exact T cell receptor, and these T cells remained in the muscle with the same characteristics. So to sum up so far, we identified muscle infiltrating T cells in 7 patients with myositis that had similar characteristics, and they recognize a mutual target per patient in the muscle. And after the treatment, these T cells remain with the same characteristics, and they are still have the same T cell receptor. So the next steps for this project was published last year, if you want to read more upon it. But we are still continuing because there's things to be understood. First, we have to expand our patient cohort. We only had 7 patients. And to be able to compare between subsets of myositis, we have to include more patients with the same subset so we can find disease-specific characteristics. And ultimately, we want to explore the function of these expanded T cells that I mentioned that recognizes a mutual target in the muscle, and whether they're pathogenic or not. And if they're pathogenic, this will allow us to have new targets to be able to develop better treatment approaches. So now, if we want to go a little bit deeper, we are going to look at the T cells that recognizes a mutual target, and then we will focus on the subset of myositis with Jo-1 autoantibodies. So, firstly, the target of the Jo-1 autoantibodies is a protein called HARS, and this is like a nice illustration of this protein. So, this protein is found in all of our bodies. They're located inside our cells. In healthy conditions, it is known that this is a self-protein, so there's no immune reaction. However, in patients with Jo-1 autoantibodies, this protein is recognized by T cells, which also leads for the antibody secretion. And the reason why this protein is recognized by T cell, there's no one single reason, but there's multiple aspects to be investigated. First of all, we have genetic susceptibility, we have environmental factors, and we have tissue damage. So the study that I'm going to talk about now, which is funded by the Myositis Association, is to understand the T cells that recognizes this HARS protein. And for this, we had to multiply these T cells because these faulty T cells that recognizes our protein are found 1 to 10 in 1 million T cells, so it's difficult to identify them without having them multiplied. So we set up a protocol in the lab where we take blood samples from our biobank and we multiplied these T cells in the laboratory. And we are looking for a T cell with a specific barcode scanner, or we can call it a lock in this case. So to be able to find a specific lock, we are using a key, and this is called a tetramer. A tetramer, you can think of it as a tool in the lab that will make us see better the lock that we want to see. And these tetramers they have a specific color. So if when this key and lock matches, it will make the T cell have a certain color, and we can find this T cell by looking at the color that it indicates. And similarly to the previous study, then we put these T cells one by one into these small vials, and then we do a genetic analysis on the T cell receptors. So our results have shown that we are able to detect these T cells that recognizes the HARS protein, which was not identified before. We are yet to understand their function, but we believe, based on the previous findings I showed as well, we believe that they're contributing to the disease progression and relapse. And here we again had the possibility to look at follow-up samples thanks to our biobank. So we wanted to see what happens to these T cells that recognize this protein during the disease progression. And from the patients, we were able to detect after 1 year of follow-up, these T cells persisted in the blood samples. So here I'm showing a patient as a example where these dark navy colored circles are the T cells from the first time point, and the squares that have the blue color are the T cells from the second time point. So if they're connected together, that means they're exactly the same T cell. So we saw that these T cells persisted. So to sum up, Jo1 specific HARS recognizing T cells were identified in the peripheral blood, and they persisted over time despite the treatment. Although we need to do more studies to understand their functions, are they protective or are they attacking the muscle or other things inside our bodies, it is possible that they are contributing to something. So the ultimate aim in my research with this antigen-specific T cells is to be able to target these faulty T cells and change their functions without destroying all the T cells in the body. This is not a near future aim or treatment, but we already have some examples of these treatments, such as the chimeric antigen receptor or CAR expressing T cells. So you might have seen there has been multiple case reports so far that these CAR T cells have been shown promising results in the patients with myositis. So what is a CAR T cell? So the T cells from the blood are extracted from the patients, and they're genetically modified to target a specific cell. And in the case of the trials published so far, these CAR T cells are targeting something called CD19. And this molecule is expressed on B cells. B cells are the producers of antibodies. So what these T cells, the CAR T cells, will do is they will recognize the B cell through this molecule, and they will eliminate them and reset this B cell characteristics in the body. So there will be no antibody producing cells anymore. So once these CAR T cells are produced, then they are multiplied in the laboratories, and then they are infused back to the patients. So so far this treatment with CAR T cell, the trials, the case reports that we have seen, they show a very good promise for inducing long-term drug-free remission in several different forms of autoimmune diseases like lupus and also including myositis. And the CAR expressing cells that have been used in the context of myositis have been typically targeting the B cells, the CD19. Although it shows a lot of promise, and there is a new possibility to for treatment of the disease. We only have a number of patients that have received this treatment so far, and there is some side effects like cytokine release syndrome or neurotoxicity syndromes. There is also the aspect of the need of lymphodepletion before the infusion of CAR T cells. So all the lymphocytes in the body has to be eliminated, and this is needed for the efficacy of the treatment. And of course, this causes possible side effects like infections, and it will also increase the necessity of hospitalization, which increases the cost of this treatment. So like the overall costs have to be considered, and we have to have more trials, more clinical trials to be able to have this treatment available for all the patients. So with that, I would like to thank everyone involved in the research that we do. Here you can see all the Myositis group, the current members and the past members, and our collaborators. The muscle infiltrating T cell project that I've shown has been led by Karin Shaman and Lina Marcelo Diaz Gallo. Team Alexandra, Merel, Anna Cristina, Juan Sebastian, and also our pathology and our flow core and our biobank and also all our founders. And for you for your attention and listening.
Well, thank you so much, Dr. Horuluoglu. That was awesome. And anyone who can take something as complicated as what you're doing and make it into a story that we can understand is clearly brilliant. So we appreciate the fact that you are really trying to understand what's happening cellularly so that someday we might have different treatments. I just want to make a comment before we get started that this webinar is specifically about research and not so much about treatments. But if you have treatment related questions, make sure you sign up for a future Ask the Doctor session. Or if you're curious about CAR T research, there is another webinar coming on the 5th of December. So I really appreciated your overview of what happens in CAR T therapy. I've worked on it a little bit in trials. I just want to remind everyone, CAR T therapy in this indication is not yet an approved treatment. It's still experimental. I think it does show great promise, and I'm super excited about it. But we need some more trials before we know if it's really something that's safe and effective for patients at large. And I want to say, of course, a huge thank you to those patients who are brave enough to volunteer to be in those trials right now. So, turning back to the research that we talked about today, what do you think it means that those T cells in the muscle are present both at diagnosis and then post-treatment? What does that mean?
Yeah, thank you for this nice question. This is something we have been uh, di- trying to discover, trying to discuss a lot in our research group. So we think the T cells are there because there's an unfinished business in the muscle tissue. Although with the treatment we can see some clinical improvements, we have seen in one of the cases that I've shown you. When the treatment dose was reduced, there was a relapse regarding the clinical manifestations. So I think we believe that these T cells are either there protecting something in the muscle, or maybe they're attacking, and we are doing more research to understand their functions. But I think it's really important to look at the muscle, look at the tissue, to understand what's going on. It's indicating that it's not gone yet. There's still something there that we need to figure out.
Actually, would you mind closing your slides so we can see your beautiful face?
Thank you.
Thank you very much. We have a great question here, and I'm a real supporter of biobanks and tissue donation, but the audience member is really curious, how do I get involved? Do I have to be in Sweden to be part of a biobank?
Well, we have a national biobank, so we have the people who are registered and living in Sweden are, if they want to be, and this is not only just myositis patients, but in all the diseases or even just healthy individuals are registered in the healthcare system that goes to these registries. And once the patients give consent, then the clinicians, the physicians can access all of their data, and if they consent, they can also donate some research samples for us. So in the context of Sweden, you have to give consent and you have to be living in Sweden. So I would definitely recommend everyone to go and register to a biobank. I mean, you don't have to donate samples, but even if you just give your information, your disease, your clinical data, this will help all of us to understand the disease, because registry-based studies are also helping us a lot to understand the outcomes of different treatments, how the patient, how the disease progression goes during the treatment, after how many years there's a remission, what happens. So it's really important that the patients also go and register for this.
And I think just adding on to that, we have some important work happening through Dr. Lisa Ryder's lab at NIH, both with patient registries and biobanking, if I'm correct. So the call to action for everyone who is a patient is actually to participate as a member of those registries, and if possible, to donate tissue. That's exactly how we enable research like Dr. Horuluoglu's and other. So if you can do it, please join that. I'm just looking through the questions here. How do I go about registering with a biobank? That's a good one.
Yes. I guess talking to your physician would be the best approach.
Probably that's a good idea. Or I think you can probably find in the United States a link in the NIH, but it might be tricky. And if Linda, if you have some advice or some suggestions, you can maybe put that into the chat for people. That's awesome. Specialty centers haven't, that's true, Shauna. I think one of the challenges, and this is a little bit of opinion, not so much fact, but one of the challenges is trying to marry all the data across all the different registries and biobanks. So I think that's one of those problems that's getting taken on by super smart people now, so that we have a broader representation of both tissue samples and data to understand both the disease and its progression and what happens with treatments, as well as what's going on cellularly.
Yeah, I agree completely. Different ethnic backgrounds, there's different progression of the disease. So it's really important to be able to combine the registries from different countries together to be able to understand the big picture.
In fact, I think I saw some research at the patient meeting this in September where maybe we're starting to identify a few patients that seem to have prolonged remission by following them longer term. So here's another question for you. What do those muscle infiltrating T cells look like in the muscles of healthy people?
Yeah, so this was something we also wondered when we found this tissue-resident memory T cells. And there is no tissue-resident memory T cells in the healthy muscles. But once we, for example, work out or like hit our arm somewhere when we have a tissue damage, there is T cells that will go travel there to see what's going on, because with the cell damage, there will be things released into the muscle, but the T cells don't stay in there. So that was the difference that we found, that these T cells that we identified, they are there to stay and they're not leaving. So that was the difference between healthy.
So those were those resident T cells that you mentioned, right? I really liked your diagram, by the way. So thank you for that. I see those little houses now in
I
my.
love. I found it recently. I showed to all of my colleagues. They also.
Um, so now you've expanded your research, you've done the work on the Joe autoantibodies. What are you gonna do next?
So I really want to understand the function of these T cells. Like I said, this is an ongoing debate that we have. Some of us believe they're pathogenic, some of us believe they're protective. It's, our research, my research is going towards the direction of understanding the interaction of the muscle and the T cell. So what are they really doing there? So this is what I want to understand further.
So when you talk about pathogenic mechanisms, can you say more about that? What does that mean in plain language?
So this T cells that I have shown with the cytotoxic function that will kill the invaders. So if the T cell is pathogenic, it is causing the muscle some harm. Maybe it's inducing the muscle to die. And also, I mean, we don't know the exact mechanism, and even if that's the mechanism, but this is some of the proposed mechanisms that they might be attacking the muscle fibers and causing the muscle damage and muscle weakness. But we don't know. We have to understand more, like how and if that's the case, how this mechanism works.
And just because I'm now very curious, have you seen any differences in those T cell infiltrations in smooth muscle versus striated or other types of muscle? Like,
Well, unfortunately, we didn't have access to different kinds of muscle tissue because the diagnosis is usually done taking from the tibialis. So we get the from the same part of the muscle. But depending on which muscle it is, there is some differences. Sometimes you won't see any T cell infiltrates, although maybe they're there, but it's just that region of the muscle that you took, it doesn't have any infiltrates. So it's a little bit heterogeneous.
and now that you say that, I don't think I really want a smooth muscle biopsy, so we'll talk about that another time.
That's what, I mean, we want to also find like some biomarkers in the blood because that will be easier if we can find something in the blood that will reflect what's going on in the muscle. So then, I mean, this is a future aim, obviously, but it will be much less invasive for the patients and easier for us as well to study.
It might even enhance the diversity of the yes. Okay, well, that's cool. I hope that happens. Um, so lots of people are, of course, are really interested in treatments and how treatments are going to evolve. Me too. But how do you think your research specifically might inform the next generation of treatments?
I actually have a nice slide to show this. I am a very visual person, so I made a let me. Yes. So I believe the research that we are doing, the translational research, the clinical research, we are building a wall where we're trying to understand the disease mechanism. So I think all the research findings that we found, it's like another brick in the wall. So we are trying to build the whole picture. It's really difficult to say from just looking at the T cells what is going on. But if we can combine the T cells with the muscle fibers, with the antibodies, with the outcomes, so then we will be able to understand the whole picture. So we believe that our research is trying to, the translational research, the basic research we do is really focusing on putting these bricks to understanding small parts. Then we can get back and look at it as a more broad picture.
I like that diagram. It really reinforces that the work you're doing at the bench and in the lab is foundational to understanding what's happening cellularly. And it will take a few more bricks before we know exactly how to build a molecule or drug that
Yes,
will affect that cellular environment.
yes, I agree. Yeah.
Okay, I'm just taking a peek here for some more questions. Oh, there will be a new research hub at NIH. Well, in fact, there are a whole lot of research hubs popping up, not just for myositis, but for other, what I'll say, big data collection efforts across disease states. And if you're comfortable sharing your data, I think it can be helpful. All right, let's see. I really liked how you explained CAR T and especially what I'll call the preparation required to go into a CAR T trial right now. And I wondered first, are any of the researchers within Karolinska participating as investigators in those trials?
Yeah, I mean, we are trying to start some clinical trials for CAR T. I don't know the details, but that is I think the future is going towards that direction. But one bottleneck in the CAR T is taking the T cells from the patients and then multiplying them in the lab. This takes a couple of weeks. So that is one of the main problems. And then there's some studies showing that we can have a biobank of T cells from healthy individuals that can be engineered to, like the ones I showed, to target these B cells and can be used. So once that is settled and that we see the effects of that, I think that will be more realistic to be able to have it, because it will reduce the costs, it will reduce the time to develop this, and it will be more available for all patients. So I think not in the very near future, but I think in the next 5, 10 years, we will see more results and like we will have more insights into it.
1000% correct. And in fact, I'm at a conference right now where CAR T and AI are very much topics of discussion. The thing that this is not related to your research, but as a researcher, I can say that when I was in graduate school, the things that we are talking about with CAR T cell therapy were only in our imagination. What is happening there is so incredibly, exquisitely complicated and beyond what I thought would ever be possible, that it's sort of thrilling. But just as you say, until we have that biobank or library of T cells where we can say this particular individual will respond to this sort of genetic modification, it's going to be hard to access both for cost and the complexity of manufacturing.
Yes. I
Yeah,
mean, even if you consider when the first CAR T cell in cancer was done, I think it was 2013. And
yeah.
after 11 years, well, let's say 10 years, then it was tried for the first time in lupus. So it is a progress. So it's not, okay, yes, we have something now, we can do it. We have to look at cost-wise as well, because it has to be available for everyone. And also without the risks or reducing the risks, having the safety, and testing it in different patients. So for now, it has been very limited number of patients that have received it.
Well, and Dr. Marks, Peter Marks, who is the head of the Center for Biologic Evaluation and Research at FDA, is very excited about CAR T for many different sorts of patients and disease states. And his words this summer were, until we can make the manufacturing process simpler, we cannot, and his word was, democratize CAR T cell therapy. So we'll put that to the side. It's coming. It won't be next year. There is a question in the chat that I will address, but very high level. There's somebody who's curious, can AI be used in research and development? And it's a yes in so many different ways, both from identifying molecules that might be helpful to perhaps modeling and predicting whether or not they will be useful and what the side effect profile will be. By the way, you do still have to test that in people, but also actually to help patients find trials and even doctors. So there's many, many, many ways that AI is going to help us. Again, I think that's a very emerging field. How have you been using it in your research, or maybe some of your colleagues are using it?
Yeah, absolutely. I agree with you completely. I mean, we have this amazing technology. Of course, we have to take some advantage of it. We have some projects using AI, mainly registry-based studies. So since we have clinical data from patients from the beginning of the disease and with different treatments and their outcomes, how the disease progressed, we can try to make models to like predict the outcomes. But I'm thinking in my projects to be able to use AI to predict these receptors that can be linked with autoreactivity. So there is some ongoing efforts to be able to include the AI into our research. And we already have we have been using some language-based models to do some predictions. Absolutely, yeah.
It's so interesting because the premise of AI is fueled on data, right? You need data to train models, to build models, and therefore, I think the cautionary aspect of AI is simply that you need good data in order to make good models. And so I guess I'm on a soapbox, but that's exactly why we need everyone to contribute to biobanks. So we have representative samples from all sorts of patients across the entire disease state.
Yes, I agree completely with you.
Um, all right. I will ask you this question, but I don't know if it's a fair one. So if you don't have an answer, that's okay. We'll put it in the list to address another time. So the question is, do T cells affect your anti-Jo numbers?
That's a difficult question. I also saw it in the questions part. So I guess it means, does the T cell that recognizes this protein affect the antibody levels? So far, I didn't see any correlation with antibody levels in these patients, but there's some studies looking at the antibody levels showing the disease severity. But in Jo-1 patients, it's a little bit unclear. So for now, we don't know, but it's something definitely that we look into to understand. I
Okay,
hope.
I think that was a great answer. I'm just taking a look now in the chat. Yeah, I think that our last call to action should be to encourage everyone who's present, whether you're a patient or a caregiver or a friend, to join TMA. This is a place where we want to share research and support patients and their families through the journey of myositis. So thank you so much, Dr. Horuluoglu. I am delighted to have met you. I hope to do so in person someday.
Thank you so much for having me and inviting me to present my research. It was very delightful. Thank you. I hope we meet in person as well soon.
Maybe next year.
Yes, absolutely. Looking forward.
And I want to say thank you to Rachel and Linda and Paula, as well as the entire TMA organization for hosting these webinars and for supporting the essential research to understand what actually is happening in myositis. All right.